So thank you for coming. I'm really delighted to be here. Mia was very, very kind to invite me, and Christian was wonderful in really putting together this lecture. So thank you so much. I want to talk to you today about our work on antidepressant responses, and I'm particularly delighted to present it here because of the long history in neuropharmacology here and how we've sort of, over the last 10 years in my lab, really tried to focus in on the questions that we've been asking. And so to sort of introduce, what is depression? And as we all know, we hear the term depression a lot, and it's used in many different ways. People saying, I feel depressed, I don't feel so great today. It's used sort of interchangeably. But as we know, it's a real medical condition, a very serious medical condition, and it's characterized by a range of symptoms. But there's not only major depression, there's dysrhythmic disorder and minor depression, of which there's many subcomponents of it. And so the point of this is really just in looking at the symptomology of major depression and thinking about this in the term of dysrhythmic, minor depression, you can see that the symptoms are a whole range of symptoms. And the fact that patients only have to present a certain number of these types of aspects makes it very, very difficult to try and model depression preclinically. How do you model an animal that's going to have these disturbances? And it's been a real challenge in the field. It's particularly a challenge when you think about what major depression is. It's a leading cause of disability in the world. The World Health Organization currently estimates greater than 350 million people are affected with depression worldwide. This is not only a toll that it takes in terms of the individual, family, and society, but it really crosses cross-culturally. In the US, I actually pulled the slide from the National Institute of Mental Health, and I like this slide even though it's a few years old for a couple of reasons. This is the 12-month prevalence of depression among all US adults by sex. Females are shown on the left, males are shown on the right. And you can see that females, it's right around 9%, and males right around like 5.3%, for an overall US prevalence of about 6.7%. And what's nice, and what I like about this slide is you can see just really how stable this is. There's not these massive fluctuations that occur with females and males. And females, as you can see, are about twofold more likely to have depression than males. The reasons for this are completely unclear. There's a group of individuals that have argued it's because estrogen makes you more susceptible to depression. There's a separate group that has argued that testosterone makes males more resistant to depression. And there's a third group that have argued that it doesn't have anything really to do with sex hormones per se. So it's really unclear, this discrepancy. Thankfully, there are treatments for depression, which we're going to talk about. The most common is treated by serotonin selective reuptake inhibitors, in which monoamine levels are raised very quickly, although the mechanism of action is really unclear because antidepressants take several weeks to work, a couple of weeks to work. It's for patients, though, that don't respond to antidepressants, of which there's a real need in the field, to really come up with a rapid, more efficacious treatment. And current clinical estimates put at a minimum of at least a third of all patients not responding to antidepressants, with some estimates even higher. And it's the ones that don't respond are the most at risk for suicide. And I pulled these numbers because I thought that they really, I think, highlight what the problem is. So in terms of the US, I couldn't find anything sort of worldwide estimates, which was what I was trying to find for this talk. But in 2003, there were 31,484 suicides in the US. To put this in some context, there were about 16,000 homicides in the US during that time frame. And it's really remarkable that when you think about US society, there's a lot of stigma that comes in terms of violence, the number of homicides that occur. But really, there's twofold more suicides than homicides. In this 10-year period, you can see that the number of suicides increased by 10,000. The number of homicides stayed relatively flat, still around 16,000. So for this huge increase in suicides, we're doing very little, really, in terms of treatment advance to treat this patient population. It's been very difficult. And suicides are something that we don't talk about. It doesn't make the news. It's sort of a taboo subject. So as I said, the most common line of treatments are drugs. SSRI is the most common line. But there's also psychotherapy, electroconvulsive therapy, which is quite safe and effective in many, many patients, and also other brain stimulation therapies that are being explored. Often there's combinations of psychotherapy and SSRIs. We first became interested in this question about a decade ago, really trying to ask, how do antidepressants work? And the caveat, as I mentioned earlier, is it's very difficult to model depression. So we really, instead of asking how do you model depression, to try to focus the question, how do antidepressants work? And as we were looking through the literature, to try to understand how antidepressants work, people take animals, they give antidepressants, primarily SSRIs currently. For a couple of weeks, they sacrifice the animals and they look for changes. What would be the genes or proteins that may be involved in mediating the effect? And there are literally hundreds, if not thousands, of changes that have been documented. But what's required? It's really hard to go from a very quick change in monoamines to a pathway in which it's going to take several weeks to exert the therapeutic effect. And there had been some work that had been done which had shown that brain-derived neurotrophic factor, BDNF, appeared to be regulated by many different classes of antidepressants. So we made some BDNF knockout mice. These were actually an inducible knockout mice where we could actually, by using a doxycycline system, could control when the BDNF was deleted preferentially in the brain, in cortex, hippocampus regions. So we did this where we could knock it out either embryonically or in the adult. And it didn't seem to matter in regards to antidepressant responses. We saw the same thing regardless of the temporal deletion. And the data is shown here. These are littermate controls, and these are the brain-specific forebrain knockouts. If we just deleted BDNF and we looked at antidepressant responses, and we did this in a range of tests, and what I'm going to just show you, most of the data for the talk is going to be the force swim test, where we take an animal, we put it in a beaker of water that's of a certain size, a certain amount of water at a certain temperature. If we take littermate control animals and we gave disipramine, and we did this with other antidepressants as well, but just as disipramine, it's just shown here, what you get is a very significant decrease in immobility, suggestive of an antidepressant effect. However, in the BDNF knockouts, just the deletion of BDNF by itself didn't change immobility, or sort of as considered baseline depression-like behavior, which we and others now have documented as well. However, when we deleted BDNF, the antidepressants didn't work, showing that BDNF was required for this effect. And it wasn't only in the force swim test, it was other behavioral paradigms that predict antidepressant efficacy, and it was with other antidepressants. So it appeared BDNF was required. So we went through a number of studies to try and figure out what brain region may be important. We're using different Cree driver lines to try and hone in on the region. And the region that really kept coming up was the hippocampus. And there is clinical data to suggest that the hippocampus may be involved in antidepressant responses. So to follow up on this, what we did is we took our floxed BDNF animals and we did AAV Cree recombinase tagged with GFP to delete BDNF in particular subregions of the hippocampus, either in the CA1 region, which is shown here, or in the dentate gyrus. And we did this bilaterally in animals. So it was a bilateral CA1 or bilateral dentate gyrus. The Cree recombinase is injected in. We wait 2 weeks so that it could actually delete the gene of interest in the particular subregion, and then we tested the animals in a range of behaviors. So what's shown on the left is the CA1 deletions. And what we found was, as expected, if we just do the control, which is just the green fluorescent protein by itself, if you give disipramine, you get a significant decrease in immobility, suggestive of an antidepressant response. If in the animals where BDNF is deleted in the CA1 region, what you see is just the BDNF loss in the CA1 has no effect on immobility, similar to the controls. And if you give disipramine, they still have an antidepressant response. So it wasn't BDNF in the CA1 region that was required. However, in the dentate gyrus, we saw something different. What we saw was that, again, in the littermate controls where GFP is just infused into the dentate, we could trigger an antidepressant effect, but this was blocked in animals where we had the localized knockout in the dentate gyrus. And we could see this in different paradigms as well as with different antidepressants as well, suggesting that BDNF in the dentate gyrus was required. And through data I'm not going to show you, it did not appear to be due to neurogenesis. We think it's the effect that BDNF can have on synaptic plasticity. But as we've been trying to work through this, BDNF, and then it's been shown through Irocastrin's group that TrkB was also required. And TrkB is the high affinity BDNF receptor. So it appears that BDNF, TrkB are really bona fide requirements for antidepressant responses in the hippocampus. But how do you go from increasing monoamines to getting to this? And this is something we've been working on for quite a while. And I think we've made some progress on this. But as we've been doing this, some clinical work came out on ketamine that caught our attention. And so this is the structure of ketamine. And these were clinical studies that were done in inpatient psychiatric units where they were treatment-resistant patients, the initial studies. And all the individuals there had failed at least 2 antidepressants in an inpatient unit. They were given an IV low dose, 0.5 mg per year, so a really low dose of ketamine infused over 30 minutes. And these treatment-resistant patients responded quickly. And in some patients, the effect was out for more than a week. Now again, because they're in an inpatient unit, they're very well known by the doctors, by the nurses. This was clearly an antidepressant response, and it didn't appear to be due to the fact that they were hallucinating or any of the other effects which can be observed with ketamine at higher doses. We were really intrigued by this data. This is one paper that came out. This was a little bit of a later study just showing sort of the time course. This was from a Carlos Zarate paper showing, again, the infusion, and in about 30 minutes of the infusion from the MADRS score, there was a very rapid antidepressant effect. Here in this study, it was more short-lived, out to about 3 days, 7 days in some. But again, there was this rapid antidepressant effect. So the field was incredibly excited by this finding, because especially in the question of treatment resistant, what do you do? Our interest came in a slightly different way. It's intriguing that ketamine can trigger a rapid antidepressant effect. The reality is that it's a single dose. It's a single dose, and we know ketamine targets the NMDA receptor. It's a non-competitive blocker. Ketamine at really, really high doses is an anesthetic, and more mid-level doses can trigger psychomimetic effects, but these were really, really low doses that were triggering this rapid antidepressant effect. And this has been replicated now in several clinical trials. Small groups, most of them have been treatment resistant, showing this rapid antidepressant effect. But there's also been a study published in bipolar patients that ketamine could trigger a rapid antidepressant effect without increasing the mania or the cycling, and also in a very small group of patients that presented in a psychiatry emergency room that were suicidal. It stabilized them very quickly instead of them having to be committed. So to us, here's a drug that targets a glutamate receptor, suggesting that it's possible to get an antidepressant effect other than through monoamines, which is the field has really been focused on for the last couple of decades. And secondly, it allows you to think about linking the receptor, the NMDA receptor, to a specific signaling pathway, as opposed to what we've been struggling with in terms of traditional antidepressants, this chronic use, and how do you get to a particular pathway. Could we link this to the pathway? So we started off doing some preclinical experiments. And the experiments are going to seem very obvious, but I think they're actually quite important. What we did is we took C57 wild-type animals, and we did a dose-response curve to ketamine to identify extremely low doses that cooperated with the human dose, per se. And we made sure that these doses did not trigger hyperactivity. It didn't trigger psychomimetic effects, no effects in like pre-pulse inhibition or learning and memory deficits, because those aren't observed in patients. So we identified doses. We then gave the animals this low dose of ketamine, and we looked in a whole range of these tests that predict antidepressant efficacy. Not only the forced swim test, learned helplessness, novelty suppressed feeding, sucrose preference, the full range. And the reason why we did this is we wanted to see if ketamine would produce an antidepressant effect in these tests. These tests are all widely used to predict antidepressant efficacy. If you give typical monoaminergic drugs that are antidepressants, they all, after putting an animal in a sort of helpless situation, stimulate the need for the animal to explore or to escape, and that's considered an antidepressant effect. So drug companies use primarily the forced swim test, but other tests like this to screen for more novel antidepressants. But it's always been unclear if you have a drug that doesn't target monoamines, would it actually produce an antidepressant effect in these tests? So here's a clinical finding. Can we use sort of a reverse translational approach to work backwards to understand the mechanism and to actually try to figure out if there's particular tests we can focus on to ask our mechanistic question? What we found was that ketamine, a low dose, worked in the majority of these tests, not in all of them, but in the majority of those tests. And one of those tests is the forced swim test, which again you see the significant decrease in immobility suggestive of an antidepressant effect. One test it didn't work in, which has received a lot of attention, is the social defeat test. The social defeat test, where an animal is put in a situation, it's beaten up. This is considered more of a model of depression per se, because chronic antidepressants will reverse the behavior. But low-dose ketamine would not reverse the behavior in this phenotype. We had to go to much, much higher doses where you have effects of locomotor activity, learning, and memory deficits. So this was not an all-or-nothing type effect and test. But we chose the test that it worked quite well in. We then said, what happens if we actually do stress animals, which is considered more a model of depression? Will ketamine still produce an antidepressant response? So we gave chronic unpredictable stress, where we did 2 unpredictable stressors a day for 3 weeks. We then injected the drug, and we did IP administration to try and mimic the more systemic effect that's seen with the IV administration. We looked at sucrose consumption, forced swim test, and novelty suppressed feeding. And what we saw is that in sucrose intake, if animals went through chronic unpredictable stress, ketamine could stimulate an increase in sucrose intake, suggestive of an antidepressant effect. In the forced swim test, again, the single dose triggered a decrease in immobility after stress, suggestive of an antidepressant effect. And in the novelty suppressed feeding, where you take the animals, you put a piece of food in the middle of a very open and bright environment, ketamine actually significantly decreased the latency to feed, suggestive of an antidepressant effect. So even under stress conditions, ketamine could produce an antidepressant effect. So with this background, we ask our first question. Is ketamine triggering its antidepressant effect through blockade of NMDA receptors? Ketamine is a non-competitive NMDA receptor antagonist. Doesn't matter if it's a competitive or a use-dependent blocker. So what we did is we took C57 animals and we gave them the low dose of ketamine, or we looked at CPP, which is the competitive NMDA receptor antagonist, or MK801, which is a use-dependent blocker. And we initially did dose response curves on these to identify, again, a low dose that could trigger an antidepressant effect without the confounds of hyperactivity or learning and memory deficits. We took this low dose of these drugs and then tested them 30 minutes later. And again, for the purposes of this talk, nearly all the data I'm going to show is going to be the forced swim test, even though we did multiple paradigms. So we tested them either 30 minutes later or in a separate cohort 3 hours later, a separate cohort 24 hours later, or in the last cohort a week later. These were all at all points different cohorts because we were worried about retesting, which could produce behavioral habituation and could skew the data. What we found with ketamine is that a single IP injection 30 minutes after produced a significant decrease in immobility, suggestive of an antidepressant effect. And in these different cohorts, ketamine produced an antidepressant effect out for a week later, which is very reminiscent of the clinical data where you can have an antidepressant effect out for a week. CPP produced a very significant decrease in immobility 30 minutes after that persisted for 24 hours, but by 1 week the effect was gone. And MK801 had a very significant decrease in immobility, suggestive of an antidepressant effect early on, but by 24 hours the effect was gone. So collectively, the data suggested to us that ketamine is exerting its effect by blocking the NMDA receptor, that it's not as simple as non-competitive, competitive, or use-dependent effects. And there are nuances to this, which I'm going to get into a little bit later. But this appears to be blockade of the NMDA receptor is triggering the rapid antidepressant effect. Now, why ketamine is the only one that produces this long-term effect, we don't know. And it's something we're actively investigating. But for the purposes of this talk, we really wanted to focus on this rapid antidepressant effect. How do you block the NMDA receptor and trigger the behavioral effect? And what's interesting to us is that all of these drugs have short half-lives, in the order of 2 to 3 hours. So how does something like ketamine, or even CPP, produce these longer-term antidepressant effects? It suggests that it's not persistent block of the NMDA receptor, that there has to be some sort of plasticity mechanism involved. In fact, clinically, this low dose of ketamine, again, in patients can produce an antidepressant effect in some patients for more than a week out, suggesting that it's not persistent blockade, and it's very unlikely even to be metabolites based on the long-term effect. So we said, you block the NMDA receptor, how do you get at mechanism? Well, as I previously shown you, we had made these conditional BDNF mice, these inducible knockouts actually. We'd also done conditional knockouts as well. And we had shown that typical antidepressants don't produce an antidepressant response. So we said, does ketamine require BDNF to trigger an antidepressant response? So what we did is we took our BDNF knockouts and we gave low dose of ketamine. What's shown here on the left is the littermate controls, and on the right here in the middle the BDNF knockouts. Again, the deletion of BDNF by itself has no effect on immobility. If we give the low dose of ketamine, you get a significant decrease in immobility, suggestive of an antidepressant response in the littermate controls that's blocked in the BDNF knockouts, showing that BDNF is in fact required for ketamine's antidepressant effect. We looked at a separate cohort of animals 24 hours later, and again, the littermate controls responded. The antidepressant effect was blocked in the BDNF knockouts. We saw the same effect when we looked at MK1. Again, MK1 could trigger this rapid antidepressant effect in the controls, but not in the BDNF knockouts. And if we look 24 hours after administration, MK1 did not trigger an antidepressant effect in agreement with our previous data, this very short-lived effect. So BDNF is required. Well, if BDNF is required, does it act on the TrkB receptor, the high affinity receptor? And so what we did is we generated conditional TrkB knockout animals where we took CAM kinase Cre that targets broad forebrain regions, cortex, and hippocampus. We crossed them with the Deflox TrkB animals. We characterized them through a range of behaviors. And what's shown here again is just the forced swim test. Just deleting TrkB alone has no effect on antidepressant responses per se, as you can see here. If we give ketamine in the littermate controls, we get a very significant decrease in immobility, suggestive of an antidepressant response, but this is blocked in the TrkB knockouts. And since the hippocampus had previously really been our focus, where it was required for BDNF and typical antidepressants, we simply took C57 animals, we gave them a low dose of ketamine or MK1, and we sacrificed them 30 minutes later, this rapid acting time point. And what we saw was that TrkB was autophosphorylated at this time point, suggesting that it was actually actively engaged. So it suggested that BDNF TrkB are required for this antidepressant effect. So how do you go from blocking NMDA receptors to requiring BDNF TrkB? So the easiest way to think about it is ketamine impacting BDNF expression in some way? So we took C57 animals and again gave them a low dose of ketamine or MK1, and we sacrificed them either 30 minutes or 24 hours later. And we first looked at BDNF message. We did real-time PCR to the coding exon of BDNF, and we looked in the hippocampus. And what we found, rather surprisingly, was that at either 30 minutes or 24 hours later, there was no significant change in BDNF mRNA expression. And these were actually large Ns, but there was nothing significant as we had really sort of expected. However, rather surprisingly, when we looked at protein at the same time points, we found that ketamine and MK1 both rather dramatically increased BDNF protein expression within 30 minutes that was back down by 24 hours. And this was rather surprising to us. This is mature BDNF. If we actually looked at the Western blots at pro-BDNF followed the same pattern, where it was up within 30 minutes and back down by 24 hours, suggesting that this could be translational dependent. We went ahead and confirmed this by ELISAs. And again, we saw this very rapid increase within 30 minutes that was back down by 24 hours. And the reason why we were really focused on this data after we got it and really wanted to confirm it with ELISAs and stuff is from some other work that had been published a few years earlier. After we had shown that BDNF was required for typical antidepressants to work in the hippocampus, there have been two groups that then took BDNF protein and they infused it either into the midbrain or into the hippocampus directly. And they saw an antidepressant effect as expected. However, what was intriguing was that they saw an antidepressant effect out 6 days after the recombinant BDNF was put in. At that point, the recombinant BDNF protein has long since been degraded, but they're still seeing an antidepressant effect 6 days later. And they couldn't really explain it. It was just sort of an interesting observation. But coupled with this data, it sort of suggested to us, is this a matter of BDNF protein can induce this sort of rapid antidepressant effect? And it doesn't have to be maintained, but it's triggering some sort of effects that are then necessary. So is this protein expression? Is this truly an increase in BDNF protein expression that's occurring as the Western blot suggest? So to do this, we looked at blocker experiments. We performed blocker experiments where we took anisomycin, which is a drug that blocks protein translation. And based on its half-life and previous work what had been published with it, we gave either anisomycin or the vehicle, then 30 minutes later gave either ketamine or vehicle, and then either 30 minutes after that or 24 hours later we did behavior. And what we saw is that 30 minutes with anisomycin than 30 minutes after ketamine. So we're at this time point. We can see that anisomycin at this early time point has no effect on immobility. Ketamine, again, by itself triggers the rapid antidepressant effect, but anisomycin blocks this antidepressant effect, showing that protein translation is required for the rapid antidepressant effect of ketamine. We see this not only at 30 minutes, but 24 hours after administration, where again, ketamine triggers the antidepressant effect, but this antidepressant effect is blocked by anisomycin. And then this is sort of correlative data. We went ahead and looked in the animals. Again, if they just had ketamine by themselves, BDNF protein was increased. But if anisomycin was present, not surprisingly, BDNF protein levels were not increased by ketamine. So protein translation appears to be required. As we went through this, though, we saw not only BDNF protein was increased, but we saw some dendritic proteins like ARC were also rapidly increased within 30 minutes. So we said it looks like this is protein translation dependent. However, is there a transcription component as well? We've looked at BDNF, we've looked at ARC, there's these proteins going on, but that doesn't mean that there couldn't be other ways in which transcription could be involved. So we decided to do these same type of blocker experiments where we block transcription. And to do that, we used actinomycin D to block new gene expression. And again, it was the same paradigm where again, we give either actinomycin D or vehicle, 30 minutes later ketamine or vehicle, and then 30 minutes or 24 hours later we do behavior. Actinomycin D at this early time point again had no effect on immobility in the forced swim test or in other paradigms. Ketamine could trigger a rapid antidepressant effect by itself, and if actinomycin D was present, it still could produce an antidepressant effect. And we see this not only at 30 minutes, but at 24 hours later. And sort of correlative data, if we have actinomycin D present, you don't see this increase in BDNF expression. So it suggested to us that this immediate effect of ketamine is protein translation dependent. Now clearly if ketamine is having effects out for a week later, there's transcription processes involved. But we're just looking at this early trigger. What happens after you block NMDA receptors? So the data so far, what I've shown you, suggests you block NMDA receptors, you upregulate BDNF protein expression very quickly. That doesn't have to be maintained, but it then activates TrkB expression. And if we think about this, there's many, many ways you can link NMDA receptors to changes in protein expression, to even synaptic changes long-term, like long-term potentiation and stuff. So there's really no surprise in that. The surprise is that when you think about all those examples, they all require activation of NMDA receptors. And this is block of NMDA receptors. So how do you block NMDA receptors and actually have this long-term behavioral effect? It's really very counterintuitive. And the data clinically has really suggested it's blockade of NMDA receptors. That's preclinically also what our data has suggested. So we ask, what happens if we do activate NMDA receptors? Do we see the same sort of effect? So we took a sub-epileptic dose of NMDA and we tested it in a range of behavioral paradigms. And what we found was that this low sub-epileptic dose of NMDA would not stimulate a rapid antidepressant effect. And not only would it not stimulate it, we could show that it did upregulate ARC expression as it's known to do, although it doesn't increase BDNF expression. So it doesn't appear that it's activation of NMDA receptors. It appears that it's block. So we went on to try and think mechanistically, how could we link our findings? And there's one theory that's kind of come out from the schizophrenia literature on how NMDA receptor blockers work. So we investigated that mechanism. And that mechanism really focuses on this disinhibition idea. And it's been done in terms of really sort of the schizophrenia literature, where they've taken higher doses of NMDA receptor blockers, primarily MK1. And when I say higher doses, we're talking in animals somewhere usually 30 to 50 mg per kg, not at the very, very low doses that are used here. So they give these higher doses. And people look by microdialysis in the cortex, and they see an increase in glutamate that they can detect in about 30 to 40 minutes after they've given the NMDA receptor blocker. So how do you get an NMDA receptor blocker and get this increase in glutamate? So what people have postulated is that you're preferentially blocking NMDA receptors on inhibitory neurons that overall raise excitation, triggering an increase in glutamate, and it's therefore through excitation of NMDA and AMPA receptors that you trigger the behavioral effects. And so there's some questions in terms of this disinhibition idea. that I've always sort of been unclear about, like why would you have a preferential effect on NMDA receptors on inhibitory neurons at such high concentrations of NMDA receptor blockers, and just sort of mechanistically linking through each of the steps. But nevertheless, it's something to investigate. Is this how ketamine is triggering its rapid antidepressant effect? We've already shown you through data here that it's not activation of NMDA receptors that is doing it. And in fact, if you ask a synaptic physiologist, they will tell you if you add an NMDA receptor blocker, you do not get activation of NMDA receptor currents. You block NMDA receptors. So it's not activation. But we said, well, what about maybe it's not activation of NMDA receptors based on this disinhibition idea. Maybe it's activation of AMPA receptors. So we gave NBQX, which blocks AMPA receptors. And when we did that, we still could not trigger a rapid antidepressant effect in any of the paradigms that we looked at. We said, okay, maybe it's not activation of NMDA or AMPA receptors. Maybe it's this disinhibition idea where you overall raise excitation. So we therefore gave a low dose of picrotoxin to overall raise excitation. And again, we could not trigger a rapid antidepressant effect. And this just shows you over here that NBQX and picrotoxin have no effect on BDNF protein expression, even though there's clearly changes that they can elicit, for example, picrotoxin on ARC, which is known to occur. So they're clearly having effects in the brain, they just don't mediate our antidepressant effect. And so to us, it suggested that disinhibition does not explain how ketamine works. There's actually been 2 follow-up studies recently, one of them done here at the Karolinska, that's actually deleted the NMDA NR1 subunit on paralbumin neurons, and ketamine still produces an antidepressant effect, again suggesting that it's not through the disinhibition mechanism. And then a second study came out a few months ago where they actually deleted the NR2B subunit specifically on excitatory neurons. And when they do that, ketamine does not work. So it suggests strongly in support of this data that it's not through disinhibition that we're mediating the effect. So it's not through disinhibition. What could it be through? Well, we ended up doing an experiment at this point that actually proved to be something really important that I'm going to come back to a little later in the talk. As I showed you, ketamine, as I've shown you before, is the only way we could really trigger this rapid antidepressant effect, ketamine or other NMDA receptor blockers. If we block AMPA receptors, we cannot stimulate the rapid antidepressant effect. However, if we give NBQX to block AMPA receptors with ketamine, we block this antidepressant effect, suggesting that while AMPA receptors are not triggering the effect, they're clearly early involved. And there's many ways to think about how AMPA receptors could stimulate plasticity, or sort of long-term type effects. So we actually ask, does ketamine trigger a sort of plasticity effect? It's sort of counterintuitive. If you take a hippocampal slice and you cut it, and you can look in the textbooks, there's all sorts of documented. It's well known that NMDA receptor blockers do not induce a sort of plasticity or potentiation. They don't. But how do you do the experiment? Well, you cut the hippocampal slice, you basically add the NMDA receptor blocker, and you record. You typically add it for 2 to 5 minutes, which is how we do our experiments, and then you record. And there's no effect. But the antidepressant effect doesn't occur in 2 to 5 minutes. So we said, what happens if we add ketamine, this low dose, for 30 minutes, which is when you start to see the initial rapid antidepressant effects. And when we do that, we actually see a very nice potentiation. So it appears there is some sort of plasticity potentiation that is occurring. That appears to be due to AMPA receptors. But again, how do you get to that point? How do you block NMDA receptors, increase BDNF protein synthesis, which can impact AMPA receptors and plasticity? It seems like there's this nice part downstream that we can start to explain, but we're still really stuck with the trigger or the mechanism. So we went through a number of different ideas of how to explain it. And to be honest, all of them pretty much fell apart until we came to the following idea. And this is some work. Ege Kavalali tomorrow is giving a talk here at the Karolinska, and a lot of this was based on his initial early work. So this is a synapse. This is sort of the presynaptic side. And we know there's 2 types of neurotransmission that occur in the nervous system. There's this activity-dependent evoked transmission, and then there's this spontaneous or sort of tonic neurotransmission. And these have been well described for a long period of time. And the work really has focused on this evoked transmission. The neuron is here, an action potential comes in, glutamate gets released, it binds to the NMDA receptor, When the NMDA receptor then is bound, calcium comes in and it activates, in particular, CAM kinase 2, which we've heard about. We know it's involved in learning and memory, plasticity, many, many different processes. What Ege's work has shown is that in terms of spontaneous, or this sort of tonic neurotransmission, the vesicles that release spontaneous neurotransmitter are different than the vesicles that release evoked transmission, such that the sort of tonic neurotransmission, when glutamate gets released, you actually bind again to the NMDA receptors. But through work that he's shown using the use-dependent effects of mKeta1, it actually suggests that with minis, with spontaneous transmission, glutamate gets released, it's actually binding a different population of glutamate receptors than with evoked transmission. And they've done this with modeling experiments using a medium spiny neuron to try and show that there's this different spatial temporal organization in terms of when you release glutamate spontaneously and activate NMDA receptors versus evoked. There's no data currently to suggest it's due to differences in NMDA receptor subunit composition. There's no data one way or the other. So with spontaneous transmission, glutamate gets released, binds to the NMDA receptor, calcium comes in again, but this time, instead of activating CAM kinase 2, you activate a different kinase, CAM kinase 3. And CAM kinase 3 is also called eukaryotic elongation factor 2 kinase. And what's intriguing about this kinase is it's only known to have one substrate, eukaryotic elongation factor 2. So when this kinase triggers phosphorylation, you immediately impact protein translation. So work by Aaron Schumann's group has actually shown that if you block these spontaneous NMDA receptors, then you inhibit the kinase, and when the kinase is inhibited, you actually don't see this phosphorylation event occurring, and you get a rapid desuppression of protein changes. You get a rapid increase in protein synthesis in about 30 minutes, which fits with our time course of ketamine. Now, all that work has been done in vitro, in primary hippocampal cultures. And they've shown that they could block spontaneous NMDA receptors using AP5 or mKeta1. It hasn't been shown with ketamine to actually have an effect on the signaling pathway. So we went in and we actually just repeated a lot of their experiments in vitro to show that ketamine could actually have an effect on this pathway. And this data just simply shows you that in primary hippocampal neurons, ketamine actually will block spontaneous NMDA receptors. If we look at the signaling pathway, if we just take our primary hippocampal neurons, let them mature, and so these are the Western blots down here, and here's the quantitation. If we look at phosphorylation of EEF2 relative to total, so if we think that spontaneous NMDA receptors are inhibiting this kinase, you therefore don't see an increase. If anything, you get a decrease in phosphorylation. We're monitoring by Western blots. We can see that if we use AP5, which is what they had previously shown, we did it side by side with ketamine. If we add either AP5 or ketamine for 30 minutes, we get a decrease in this phosphorylation of EF2 to total, suggesting that the kinase has been inhibited. If we look 2 hours out with application of either AP5 or ketamine, again, we see a decrease in phosphorylation of EF2 relative to total. Work that Aaron and Mike had done had previously shown that if they add TTX, which blocks evoked transmission, the other form, that it doesn't impact the signaling pathway. And they argued that it's because that they are separate pathways. And in fact, if we took TTX, we didn't see any effect on phosphorylation of EEF2 to total. It's not inhibiting the pathway. It's not inhibiting the kinase. We went ahead and added TTX with either AP5 or ketamine for 30 minutes or 2 hours later. And we saw, again, a similar decrease in phosphorylation of EEF2 relative to total compared to just AP5 or ketamine alone, suggesting again that there's not crosstalk between the pathways. So this is all in vitro. So what about in vivo? Does this explain how ketamine works? So we took C57 animals, we gave them a low dose of ketamine or mKeta1, and we looked at phosphorylation of EEF2 relative to total. We did this first by fluorescence, and you can see that ketamine actually triggers a decrease, that's shown here by the brightness, of EEF2, phosphorylation of EF2 relative to total per se, which is shown here by Western blot when we quantitated it. So we quantitated it by Western blot as well as by immunocytochemistry. So it appears that we are blocking spontaneous NMDA receptors to inhibit this kinase to trigger the effect. So if we're right, which is our hypothesis based on this data, then if we just inhibit this kinase alone, could we trigger a rapid antidepressant effect, independent of blocking NMDA receptors? So we took two drugs that are EEF2 kinase inhibitors, Ritalin and NH125. They both will inhibit EEF2 kinase, but like most kinase inhibitors, they can also inhibit other kinases as well. So they're not the most selective, but this was our starting point. So we took Ritalin and NH125. We did dose response curves with them. We first looked to see what happens to BDNF protein expression. And both Ritalin and NH125 produce a significant increase in BDNF protein expression within 30 minutes in the hippocampus following administration. And in those same samples, they trigger a decrease in phosphorylation of EEF2 relative to total, which you would expect if they're inhibiting the kinase. When we looked behaviorally, in C57 animals, we saw that Ritalin and NH125 both could produce a significant decrease in immobility 30 minutes after administration, suggestive of a rapid antidepressant effect that was present in different cohorts of animals 24 hours later, that was present at least with Ritalin a week later. So it suggested just inhibiting the kinase could trigger a rapid antidepressant effect. Does this tie into our mechanism? So we took our BDNF knockout animals, which I'd previously shown you. Again, just deleting BDNF has no effect on immobility, suggesting no change in sort of antidepressant or depression-like behavior per se. Ritalin, again, produces a significant decrease in immobility, suggestive of an antidepressant effect that's blocked in the BDNF knockouts, suggesting that EEF2 is upstream of the BDNF-dependent effects. So we said, okay, this is wonderful. However, the biggest caveat is that Ritalin and NH125, while they inhibit EEF2 kinase, they also could be working through other kinases as well. So we obtained the EEF2 kinase knockout animals. We characterized them through a range of behaviors. Just the initial characterization in terms of ketamine, this just shows you that these kinases, as expected, do not have any phosphorylation of EEF2. If we take littermate controls or knockouts, if we give them ketamine, as shown in black, again, you get this rapid increase in BDNF protein in the wild types that you don't get in the knockouts. And behaviorally, the EEF2 knockouts have no change in immobility by themselves in the Force-1 test. The littermate controls respond to ketamine, but it's blocked in the EEF2 kinase knockout animals. We see this not only, like I said, with the Force-1 test, we did a range of behaviors. This is the novelty suppressed feeding test. Again, ketamine in the wild type animals triggers a significant decrease in latency to eat, suggestive of an antidepressant effect that's blocked in the knockouts. So EEF2 is required for ketamine's antidepressant effect. It appears to be upstream of BDNF. So our data really suggests that we're blocking spontaneous NMDA receptors. We inhibit this kinase. We desuppress protein synthesis. And that, it's been shown BDNF can actually have effects on plasticity. And we'd actually, as I showed you before, have this sort of potentiation occurring. So we came back to this potentiation. And we said, is this potentiation, it seems to be correlating with our antidepressant effects. But what is the mechanism? I mean, it's really surprising that ketamine, an NMDA receptor blocker, is going to have this rapid antidepressant effect that's going to persist for quite a while. It suggests, again, that there should be some type of plasticity. But since it's an NMDA receptor blocker, what can we say about it? So we first took our EEF2 kinase knockout animals relative to littermate controls. Again, with the littermate controls, we cut our hippocampal slice, we add ketamine for 30 minutes, wash it out, and we see this very nice potentiation. But this potentiation is blocked in the EEF2 kinase knockout animals. It's also blocked in the BDNF knockout animals. Again, in the littermate controls, we see this potentiation, which we don't see in the BDNF knockout animals that also don't respond to ketamine. This potentiation is protein translation dependent. If we had ketamine in the presence of anisomycin, normally we see this potentiation that would be up here, but it's blocked in the presence of anisomycin, just as it does with mKeta1 and anisomycin. And I think in some of the most provocative data that we have, if we take ketamine and apply it to our hippocampal slice in the presence of TTX to block evoked transmission, we still see this potentiation. Again, arguing for the separation of the pathways. And if we just add TTX by itself to block evoked transmission, we don't see this potentiation. If anything, we see a decrease. So with that, we went on to look at this plasticity a little bit more. primarily because there's been a lot of talk within psychiatry about the role of AMPA receptors in the ketamine effect. And our data clearly shows that this potentiation appears to require AMPA receptors. So does it require AMPA receptors more directly? So what we did is we took the EF2 kinase knockout animals, which were required for the antidepressant effect, the potentiation effect. We think this is the point at which is necessary for the ketamine's rapid effect. If we take littermate controls and give them ketamine, and we look at surface expression level, these are biotinylation experiments of GluA1, we can see that ketamine triggers a significant increase in surface expression in the wild-type animals, but not in the EF2 knockout animals. And the same thing in terms of GluA2. We see a significant increase with ketamine in GluA2, but not in the EF2 knockouts. And the GluA2 was a little bit surprising to us. We saw the same effect with the BDNF knockouts, and we see an increase in GluA1 and GluA2 in the wild types, but not in the BDNF knockouts. We looked at this electrophysiologically. Again, ketamine will trigger this potentiation. If we did ketamine in the presence of NASPEN, that blocks GluA2 lacking receptors, because GluA2 is really the surprising part here in terms of this potentiation. So if we add a drug that blocks GluA2 lacking AMPA receptors, you still see this potentiation. However, if we add DNQX that just blocks AMPA receptors in general, we block this potentiation. We went ahead and obtained the GluA2 knockout animals, and you can see that in the wild-type animals, you have this potentiation present that's blocked in the GluA2. So GluA2 clearly was required for this potentiation. It's not just GluR1 dependent. Moreover, when we took these animals and we did behavior, in the littermate controls, ketamine could trigger a rapid antidepressant-like effect that was blocked in the GluA2 knockouts. This is the forced swim test. This is the novelty suppressed feeding. Again, the decrease in latency, which was observed in the littermate controls with ketamine, but not with the GluA2 knockouts. So our data at this point really suggested the following model. That what we believe is that ketamine, this really low dose of ketamine, we think the dose is critically important. It's not all doses of ketamine. Ketamine at high doses triggers anesthesia, more mid-level doses, psychomimetic effects. These are the really, really low doses. The fact that you get these very different behavioral effects, it's unlikely that the mechanism of action at these different effects are going to be the same. So we think that ketamine, really low doses of ketamine, are blocking spontaneous NMDA receptors, which inhibit the kinase such that you don't get this phosphorylation event, desuppressing protein translation, including BDNF, which is required for the antidepressant effect, which then triggers insertion of GluA1 and GluA2 for this longer-term effect. So I want to just in the last couple of minutes, just very, very quickly, try and tell you why I think that this is important. This is our mechanism, and it's a testable hypothesis. And we're doing a lot of work on this, trying to look at this in terms of what it means and what it does. But let's come back clinically. When ketamine came out, it triggered a great deal of interest and excitement. But the problem was because ketamine could have a number of side effects. You don't really want to be prescribing ketamine wide range. There's a lot of studies going on trying to right now look at potential dosing of ketamine. What happens if you give one dose and then maybe a few days later give another low dose? What happens if you give a low dose of ketamine and then you try a patient on an antidepressant or maybe ECT, would they suddenly now maybe respond? People are looking at patch for ketamine, like sort of a patch administration or maybe intranasal. There's a lot of work going on with it, but some people are really, really hesitant with ketamine. They're worried about the side effects, the abuse liability. So a number of people, clinicians, said, okay, the way to circumvent this is let's take memantine. Memantine is also a non-competitive NMDA receptor antagonist. It's FDA prescribed for Alzheimer's, and use that. Does that trigger a rapid antidepressant effect? And so, as I said, it's an NMDA receptor blocker. It actually has lower affinity for the GluA2A, 2B isoforms than ketamine in the presence of magnesium. So even though they both are non-competitive NMDA receptor blockers, there are differences between the two. What's been found now in 3 trials is that memantine does not trigger a rapid antidepressant effect, and even out where people have actually prescribed it out to 8 weeks, there's no antidepressant effect that's been observed. And there's been a range of doses that have been looked at. So clearly, there's something different between memantine and ketamine. And again, there are clearly differences that are known, but it's not a simple non-competitive NMDA receptor antagonist that's going to produce a rapid antidepressant effect. So we said we have a hypothesis. Does this hypothesis play into the different action of how memantine and ketamine work, since one triggers a rapid antidepressant effect and one doesn't? So very quickly, I'm just going to go through this last part very quickly, but the bottom line here is that when you actually look at the conductance versus the holding potential, what you see is in the presence of magnesium, in particular this 1 millimolar, which is physiologically relevant, there's a residual current that occurs with the NMDA receptor at resting membrane potentials. And as I've shown you, this component here we think is really critical for ketamine's action. We know that if we take memantine, again, doing dose-response curves, trying to sort of hit the low doses as well as the doses that have been tried clinically, that it does not exert rapid antidepressant effects in animals, similar to the clinical findings. This just shows you locomotor activity. Memantine has very little locomotor effects. The low dose of ketamine does have some locomotor effects very early on, within the first 5 to 10 minutes or so, but when we do our testing at 30 minutes, the effects are gone. When we look at the forced swim test, we looked at 30 minutes again, and memantine, we looked at 3 different doses of memantine, and it did not exert an antidepressant effect, where side-by-side ketamine did. We said, okay, perhaps the memantine effects are delayed, even though clinically it doesn't work. So we looked at 8 hours and 24 hours in separate cohorts, and again, the memantine did not trigger antidepressant effects. Also, it didn't trigger antidepressant effects in the novelty-suppressed feeding. So this is similar to the clinical finding. So we did electrophysiology, and what we're trying to do really quickly is just block all types of receptors and isolate the NMDA receptor component. So we simply culture primary hippocampal neurons. We're going to patch the cell. We're going to record our minis, looking at the spontaneous NMDA receptor component, if you will. We'll then perfuse on in our NMDA receptor antagonists, either ketamine or memantine, and then we're going to record. So this is in the presence of no magnesium. So under these conditions, this is sort of like what would be mimicked is when the cell is sort of depolarized, if you will. NMDA currents are really pronounced. And when we do that, ketamine and memantine look very similar. When there's no magnesium present, they both work on NMDA receptor-mediated synaptic currents very, very equally. We then said, what happens, though, under these conditions where we're looking at the sort of resting membrane condition, if you will, with physiological magnesium? And so we did the same type of paradigm here, but this time we actually added physiological magnesium. So what we're doing here is we're recording AMPA and NMDA receptor currents, and then we're testing the ability of either memantine or ketamine to block the NMDA receptor component. So basically we're using the AMPA receptor current as a tag, if you will, to see where to look. And these are whole cell patch clamp recordings, and this just tells you that the NMDA receptor component of these miniature EPSCs is small, but it is present, so we can detect it. And so I'm not going to go through this in great detail, but the bottom line is under physiological magnesium, ketamine blocks the NMDA receptor, but memantine does not block it. So there is a functional difference with physiological magnesium to the NMDA receptor. So we said, does this extend to the intracellular signaling cascade that we proposed for how ketamine works? So we simply looked at memantine's ability to alter phosphorylation of EEF2 relative to total EEF2 in comparison to ketamine. And ketamine, 30 minutes after administration, it triggers a rapid antidepressant effect. It also triggers a significant decrease in phosphorylation of EEF2 that we don't see with memantine. We looked to make sure that this wasn't a delayed effect. We looked 8 hours and 24 hours later, and we didn't see it. And you can see that the ketamine effect, again, this effect on signaling is very rapid. It doesn't have to be maintained. It's just sort of the trigger point to mediate the effects. We also looked at BDNF expression. Ketamine, again, introduces this rapid increase in BDNF protein in the hippocampus that, again, doesn't have to be maintained, but it is a rapidly induced that's necessary that we don't see with memantine. So our data suggest that ketamine, by blocking spontaneous NMDA receptors and inhibiting this kinase, triggers this desuppression of protein translation that is necessary for the rapid antidepressant effect. Memantine, under physiological magnesium, has a different effect. Its effect on evoked transmission is very similar to ketamine. But when you're looking at spontaneous transmission, the sort of resting transmission, if you will, there's a different effect. And this is the only hypothesis that I know that's been put out that can maybe explain the difference between ketamine and memantine, of why one works and why one doesn't. Why I think that's important is that right now, especially clinically, there's a number of drugs that are just being tried in patients, and they're in general not working. So could you try to use this mechanism to perhaps screen for drugs that might actually have a rapid antidepressant effect? That's one idea. So what are we doing with this? Showed you a lot of data. Well, we're concentrating our efforts on a number of different components of this. But in general, one thing what we're looking at is this downstream effect. We're trying to understand why ketamine can still have an effect in some patients out to a week later. What happens after this TrkB activation? We're also doing a lot to try and understand this plasticity, this potentiation. What is this good for? It's sort of, if you will, a homeostatic plasticity mechanism. People like Gina Taragiano and others have shown that when you block this sort of NMDA type current or use TTX, you can actually get sort of a rebound effect. But does this occur in vivo? How it's really been shown has been through a lot of very sort of severe surgical type procedures or something pretty blatant. But could ketamine, that's working as a drug, very rapidly be inducing a sort of homeostatic plasticity mechanism? So we're doing a lot of work to try and understand that. We're also focusing a lot on BDNF TrkB. Where is our BDNF being made? Where is it being released? Is it anterograde, retrograde, presynaptic, postsynaptic? There's a lot of components of this that really are unclear. We're also looking upstream. If we're correct about this kinase being so important in terms of disinhibiting it, that you don't get this phosphorylation event that really sets this pathway in motion, there should be a phosphatase that balances this reaction. And it's unknown. We've identified the phosphatase recently in lab, and it's through ongoing work right now, which is why I have not presented it. If we inhibit the phosphatase, we interfere with ketamine's ability to trigger a rapid antidepressant effect. And what's interesting is that the phosphatase, how it's regulated through our early data, there's particular drugs that could infect it. And some of those are used clinically. And there has been some work showing that if patients are on this certain class of antidepressants, they might not respond to ketamine. So we think it actually does play out. So we have to do more work on this. But then we're also trying to look presynaptically. If we're right and that this is actually spontaneous transmission that mediates the effect, can we actually go in and block spontaneous transmission in vivo? If we do that, would it impact ketamine's ability to trigger an antidepressant effect? If we can actually show that, and we can actually manipulate this is a collaboration with Ege Kavlavi, where we can manipulate spontaneous transmission in vitro selectively. Now we're trying to take this in vivo. If we can do that, it allows us to ask the question, what is the role of spontaneous transmission in vivo? Does it impact learning and memory? What sort of processes could be involved? So these are ongoing studies in my work. I want to just conclude. This is just the building where I'm at. UT Southwestern is much bigger than this, but this is just the building where I'm at. This work, these are the individuals, the work that I presented. Anita Autry did a lot of the initial behavior, as well as Kristen and some of the follow-up studies that I presented today. Megumi Adachi was involved in a lot of the biochemistry type work, as well as some of the behavior. She was really instrumental in many aspects. She was a postdoc in the lab. Erin Gideons did the memantine study. She's a leaving graduate student right now. Lena did the potentiation experiments that I showed. She's working in collaboration with Ege Kavlavi. Pingfei Chen is a statistician that we've worked with for years. And Alexei Reznov was kind enough to provide us with the EF2 kinase knockout animals. So with that, I will stop, and I'm happy to answer any questions.